Introduce our first speaker of the session, Pedro Clavel. He is an undergraduate student in Brian Phillips Lab at the National Institute of Pediatrics, and he's doing very fascinating work studying metabolic syndrome effects on olfactory function, memory, and neurodegenerative process in rat. And they recently published a related paper about metabolic syndrome that you'll be able to find the link to it in the chat. And finally, very exciting that Pedro, he's doing science communication. He's a part of a Spanish spoken science communication podcast. And Justo un Medio, I apologize for my Spanish. And you'll in the chat, and you can also follow him on Spotify. So with that, Pedro, welcome, and please go ahead.
Thank you very much, Kathy, for the presentation. So let's begin. This is the title of my project, Effect of the Metabolic Syndrome on Olfactory Function, Memory, and Neurodegenerative Process in Rats. So Alzheimer's disease is a neurodegenerative condition characterized by a cognitive decline, mainly in memory function. There is evidence that shows that olfactory impairment, such as anosmia or hyposmia, is present in Alzheimer's disease patients even years before the onset of classical symptoms as memory loss. Neuritic plaques and neurofibrillary tangles are the hallmarks of the amyloid precursor protein drives the neuritic plaques formation in the. This is favored by the metabolic syndrome, which is a cluster of metabolic disturbances such as obesity, and specifically its core dysfunction and oxidative stress. So let's review more of that. The evidence shows that it promotes the amyloid pathway processing of the APP in neuronal cell cultures. In Alzheimer's disease transgenic models, a high-fat diet feeding, or a streptozotocin injection promotes the amyloid beta peptides accumulation, as well as overexpression of base-1, beta-amyloid cleaving enzyme, and full-length APP. Thus, this evidence shows that insulin impairment is present in the development of neuritic plaques. The evidence shows that amyloid beta peptides in the olfactory epithelium deposition is present at early stages of Alzheimer's disease patients. There is also evidence that shows that oxidative stress is present even months before the deposition of neuritic plaques in an Alzheimer's disease transgenic model. So part of this previous evidence comes from diabetic models with impaired insulin and glucose metabolism. But there is insufficient data whether if these alterations occurs in a metabolic syndrome model induced by a high carbohydrate diet, which exhibits insulin resistance, but glucose metabolism seems to be unaffected. So, I have some problems. So our hypothesis was that metabolic syndrome will generate behavioral alteration similar to those observed in Alzheimer's disease in olfactory and memory function, as well in its neuronal and molecular substrates. Our main objectives was to evaluate if there are alterations in olfactory and memory function in a model of metabolic syndrome induced by a high carbohydrate diet, and to assess whether insulin resistance and/or oxidative stress alter the proteins involved in the formation of neuritic plaques in the olfactory bulb and the hippocampus. So for this investigation, we use male Wistar rats divided into two groups, a control group that received standard chow plus water ad libitum, and an experimental group which received 30% sucrose in their drinking water. This treatment lasted for 23 weeks. Olfactory and memory function were evaluated on week 21 and week 22. At week 23, somatometric and biochemical parameters were measured, and sample recollection was performed after euthanasia. Olfactory bulb and hippocampus were dissected to perform the following techniques: Western blot analysis, MTA determination, and histological analysis. So rats that receive sucrose in their drinking water develop metabolic syndrome, since final weight, triglycerides, and adipose tissue were significantly increased. So this model is ideal to the early metabolic disturbances, since insulin resistance is present, as seen as in a hyperinsulinemia state, but glucose remains unaffected. Olfactory function was evaluated using the habituation and dishabituation task and the varied food test. In the former, rats are presented with consecutive olfactory stimuli. Habituation is defined by a diminished olfactory exploration for the same odor, and dishabituation is defined by an increase in olfactory exploration in the presence of a new odor. So both groups exhibit habituation and dishabituation. Both metabolic syndrome rats had a trend to shorter exploration times. In the varied food test, rats with metabolic syndrome took longer to discover the hidden piece of food. Thus, this evidence shows that metabolic syndrome rats have olfactory impairment. Memory function was evaluated using the novel object recognition test. In this task, rats are familiarized with two identical objects for several trials. A day later, one of the objects is replaced by a new one with different characteristics. So both groups prefer to explore the novel object more than the familiar object. Both rats with metabolic syndrome had shorter exploration times for both groups. Despite of that, rats with metabolic syndrome prefer to explore the novel object. So these results seems to indicate that rats with metabolic syndrome had a memory improvement. Next, we evaluated the APP and base-1 expression and insulin pathway activation in the hippocampus. Insulin pathway is hyperactivated, as seen as in higher phosphorylation levels of insulin receptor and AKT protein, together with lower expression of the APP protein and a trend to lower base-1 expression. So insulin pathway hyperactivation might account for the apparent memory improvement seen in the rats with metabolic syndrome. Nevertheless, none of these changes in protein expression and protein phosphorylation were observed in the olfactory bulb. So we propose that olfactory bulb might be one of the first brain areas to exhibit insulin resistance, since insulin receptor remain unchanged despite systemic hyperinsulinemia, in contrast to what we found in the hippocampus. Oxidative stress was evaluated using by lipoperoxidation, measured using malondialdehyde levels. MDA levels were lower in rats with metabolic syndrome in their hippocampus, and tended to be lower as well in the olfactory bulb. Thus, likely, the metabolic syndrome likely produced an antioxidant response. Finally, we evaluated neurodegeneration was determined using the fluorogate C staining in hippocampus and olfactory bulb. No apparent signs of neurodegeneration were observed in the hippocampus, and despite a trend to higher signal of this staining, the data dispersion was too high, so we couldn't find any differences in neurodegeneration markers in the olfactory bulb. So as a conclusion, we provide novel evidence that shows that olfactory impairment is present in rats with metabolic syndrome induced by a high carbohydrate diet. Memory was appeared to be improved in these rats, likely due to an hyperactivation of the insulin receptor, insulin pathway. And this together with lower APP expression and MDA levels. None of these changes were observed in the olfactory bulb. So we propose that the olfactory bulb shows early insulin resistance, and this could account for the olfactory impairment seen in these rats. So on insulin pathway impairment and amyloid beta deposition, accumulation in the olfactory bulb merit further research. So finally, I want to thank everyone involved in this investigation, to my lab, the Laboratorio of Nutrición Experimental, as well as Laboratorio de Neurociencias and Laboratorio de Biología de la Reproducción from the Instituto Nacional de Pediatría, and I want to thank my friends. So that's all for me. I'll do my best to respond all of your questions.
All right, thank you very much, Pedro, for a wonderful talk. And it's a very interesting and comprehensive study that gives us some insights into the link between metabolic syndrome and olfactory dysfunction and Alzheimer's disease. So thank you again. And I encourage everyone to please ask questions.
So we have a few questions on YouTube.
Okay.
So I'm going to read it. Cynthia has asked, can you speculate why the imbalance on diet have an effect on olfactory function?
Yes, because the hyperinsulin, the insulin pathway is involved in many of the neuronal process as synaptic strain, protein expression that forms the synapses to form the synapses between the neurons. So we, as well as clinical evidence shows that metabolic syndrome and obesity have this olfactory impairment. We wanted to study the correlation between this alteration and the Alzheimer alteration that have also as well olfactory impairment. So I don't know the causal link, but we established that maybe insulin activation, insulin pathway might account for this olfactory impairment, but we didn't evaluate other options like free fatty acids or the influence of immune cells. But yeah, we found an apparently insulin resistance in this area, in the olfactory bulb, and so we propose that this is implied in the olfactory impairment.
Thank you. She continues, sometimes it is speculated that olfactory malfunction predicts neurodegenerative diseases, yes, but you show maybe before something at the metabolic, so you show something even before at the metabolic level is happening.
Yes,
so
yes, yes, olfactory impairment is being proposed as an early biomarker of neurodegenerative diseases, and because the interrelation between insulin pathway and the development of the Alzheimer's disease hallmarks as neuritic plaque formation. So we established as well as that this interrelation could be important to study these alterations.
Thank you. I hope she writes back if the answer was enough or she needs more.
So, yeah.
Okay, thank you.
Can you hear me?
The questions.